Hello everyone, myself Revati. Today in this video we are going to study the how probability is used in genetics. So it is one of the application of probability. So you all have knowing the definition of probability. Probability is nothing but prediction. Probability is the likelihood of something happening. We are predicting whether it will rain today or not, so we are predicting. Probability is equal to possible choices divided by total number of options. So when I toss a coin, we will get two possibles like head and tail. So there are two possible. Out of two, anyone will get whether head or either tail. So this is known as simple definition probability. So how probability is used in genetics? So Mendel was the first scientist to determine the probability is used in genetics. Mendel was the first person to realize that probability can be used to predict the result of genetic crosses. In other words, he could use probability to know all the offsprings of the first generation would be tall without even seeing them. So by this we come to know that by crossing the two breeds, what are the possible of getting different genes with different characteristics. So by without seeing, he used the method called probability. So whenever we cross the two plants, it will get a gene from both a mother and other father. So how, what are the possible, what characters did the gene offspring get from his mother or a child? For this he used the probability. As we know that probability theory, we know come to know the many definitions like probability is how it is used in intersection and union. There are three types of classical approach: frequency approach and axiomatic approach. Measurement, this is the three measurement approaches construct a measure of probability. Exclusive events, we this all with this definition we studied in our theory classes. Favorable events, mutually exclusive events. So this is the fundamental definition in mathematics. This is the same method how he is going to apply it. Before going, these are the some of the genetics important features. Phenotype, the physical appearance of the offspring. So whether a child how get tall or short, every child is getting genes from their elder ones like their parents. So tall, short, this physical appearances is called phenotype. The genetic makeup or allele combination of the spring, and this genotype cannot be seen by the physical. So next definition is homozygous. Two of the same alleles like both capital T. Both capital T represents the dominant tall and both small t represent the dominant tall, and this is recessive tall. Heterozygous, the hetero means different genes. One allele contains different genes like capital T and small t. These two are different genes. So these are the basic definition in probability. So he Mendel used one mathematical approach is Punnett squares. Punnett squares is nothing but the graphical representation of predicting the number of possible genes. So when two parents are crossed, what are the possible of getting only head, only tall, only short? What are the possible of getting both short and tall? So by this he used to predict the Punnett square is nothing but the graphical representation used by the Mendel to predict the gene. So it can be used in various diagram that is used to predict the genotypes of a particular crosses, and this we in this analysis we get the correct results. The probability of an offspring having a particular genotype can be recommended. So this Punnett square helpful tool that helps to predict the variations and possibilities that can from crossbreeding. This includes predicting crossing plants, animals, even humans with each other. So it is nothing but there are two genes. Consider like tall and short. When these are crossed, and these how what are the possible of getting the accurate gene? This is the example. Determine the expected percentage of different genotypes in the offsprings of two parents. When we cross the two parents, then only we can come to know the what are the possible of chances that this particular genes will contained by the offsprings. So, an example of Punnett square for a pea plant. As we all studied in our 10th classes, pea plant, Mendel is the first scientist. He crossed the first plant is known as pea plant. He has crossed. In this example, both parents are heterozygous and some are homozygous. As I already discussed, homozygous is nothing but a both the genes containing same like tall, and this that particular offsprings must contain only gene from the father, that is not, but that is must be containing same, that is known as heterozygous condition. Heterozygous is nothing but a offspring can contain a gene from either both the parents like mother and father. This is known as genotype of a offspring. So this genotype can helps to solve the more problems. So let us consider example of Mendel crossed two genes like capital T, small t, and how it is crossed TT. This T, small t, is nothing but recessive, and this capital T is nothing but dominant. When both dominant and recessive contains, then that gene is having the more one having the highest precedence. So small t and capital T, this the offspring must having the higher chances of capital T, means the offspring must be high, which is dominant, that will be going to consider. And this is small t, so small, both are small t means, and that offspring must be small. So this is the simple example. 3 is to 1 and 2 is to 1. There are many example like 2 is to 1, 4 is to 1. Many example, whatever with that, when we cross the two genes, we construct a simple Punnett square to get to know the accurate result of the offsprings. Here both are capital T. This is dominant tall, and this one gene is having recessive and another one gene is having capital T, is nothing but the one who is having the dominant can take place the result. So this is the just for pea plant. We not only for pea plant we have going to cross. There are many different genes we are going to cross. By considering the, we must consider the characteristics. For example, this T and this T. This we have took an example of phenotype. As I already discussed in the previous slide, phenotype is nothing but the physical markup of a genes. This short, tall is a physical markup of a gene. So the potentially the in when we cross the 2 is to 1 pea plant, we are getting a number of possible of getting only capital T is 1 by 4. So there are 4 chances of getting possible, but only getting capital T, that is pure tall plant is called pure breeding, is called cross breeding. So probability in we have studied about the in theory classes like multiplication rule and addition rule. Multiplication rule is nothing but two events occurring at a same time. There must be two events occurring at the same time is known as multiplication rule. And addition rule, one event can occur in both different way, and this is the only particular event occurring in different ways. This is known as Addition rule. Before going to start the event, first we know the definition of events. In probability theory, an event is a set of outcomes of an experiment to which a probability is assigned. So, as I told, when I toss a coin, there are two possible, either head or either tail. This two possible is nothing but a event. This is the event, and this multiplication and how to apply in the problem, that is very trickiest question in the probability. When to apply addition rule, and when to apply the addition rule and multiplication rule. According to the multiplication rule of probability, the probability of occurrence of both the events A and B is equal to the product of probability of B occurring and the conditional probability that event A is occurring at the event B occurs. So this is the probability of the simultaneous occurrence of two events that are independent in which by the product of their individual probability. So this is very important. This multiplication can be ruled when there will be a individual probability of occurrence will be there, and the multiplication rule must be used. So multiplication rule and addition rule are nothing but the how we are going to apply the problems, that is very important. That we use for computing the probability of A and B as well as probability of A and B for two given events A, B defined on the sample space. Sample space is as I told, the number of problems whatever we are given, that is how we are going to apply, that makes the difference in the probability. So we all have studied about this is about the multiplication rule and addition rule. One event can occur in both different ways. Only one event can occur in both different ways. See, let us take an example. The couple has 3 children. So the couple has 3 children. So according to the question, we have to the question must be at least they contains 2 daughters and 1 son. More than that, it is not possible. We have to use this condition and construct the probability. So already we have to include 2 daughters and son. So first there will be a chances of first 2 daughters and after son. First son and after there will be a chances of 2 daughters. And first there will be a chance of daughter, son, daughter. So in generally, the couple must produce either daughter or other son, that is half, and producing the son is also half. So let us consider the first question, daughter, son. Take the term individually. Listen to the question, take the term individually. So probability of occurrence of daughter is 1 by 2. Probability of occurrence of daughter is 1 by 2. Probability of occurring son is 1 by 2. So here we have to use multiplication. Why means we have taking the term individually. While I am explaining the multiplication event, I told we are used to multiply the individual term when there must be use of multiplication rule. Second is son, daughter, daughter. This is also same, and this will get 1 by 8, 1 by 8, 1 by 8. And this in all three cases we get a 1 by 8. And what must be do that 1 by 8? 1 by 8, then this is the 2 daughter plus son occurring in more different ways. So we have to add. See, more different ways means daughter, son. This is the only one event occurring in more different ways. So we have to use addition rule. 1 by 8 plus 1 by 8 plus 1 by 8, that is equal to 3 by 24. So what is the question? What is the already the couple has 3 children, and there must be 2 daughter and 1 son. So the probability of what is the probability of having 2 daughter plus son? So the probability of having 2 daughter plus son a couple is having is equal to 3 by 24. So this is just one simple example of probability. So now let us learn about the how addition rule and exactly helps, and these questions will solve by many we will get to know in bank exams, post exams. Then this is are the general questions: when to apply the multiplication rule and when to apply the addition rule is most important. The question may be changed. If the question may be changed, two sons and one daughter, we have to adjust in three childrens only. We cannot take two daughter plus two sons. That is not possible. According to the condition of the question, we have to adjust. Suppose there must be a we must think the question like as I already told, a couple has three children. Both the children may contains daughters and three of the children may contain son, but the question given the condition is two daughter plus son. According to the question, we have to adjust our rule. So this is the just example that I have shown the addition rule and multiplication rule in probability. So 3 by 24 is the probability of getting two daughter plus son. So let us take another example. When as I already told, when two genes are crossed, we get a one new offspring. So when capital A, two capital A, capital B, small b, capital C, small c are crossed with capital A, small a, capital B, small b, capital C, small c, we get a result of capital A, capital B, capital B, capital C, small c. So how we are going, what is the probability of getting this gene? When these two are crossed, what is the probability of getting this gene? So this is the one different example. A and we take the first order, that is 2 capital A is crossed with 1 capital A and small a of different gene. When by the help of Punnett square, so this is the one of the application of Punnett square. Cross it, when A is crossed with capital A, small a, A, and capital A, small a we get. But what actually we want? We want capital A and small a. How many times the capital A and small a is repeating? So it is 2 by 4. Why 4? There are total 4 possible, but we are getting capital A and small a is 2 times. So 2 divided by 4, 1 by 2. Similarly, when capital B and small b is crossed with capital B and small b, we get the result of both the gene contains both capital B and capital B. So when it is crossed capital B, small b, small b, capital B, and both are small b. Here also the total number of possible of events is 1 by that is 4. And what are the chances we get? We get only capital B. So it is occurring how many times? 1 time. So it is number of possible divided by number of choice. What choice we have? That is capital B. 1 divided by 4. So next consider a third. When capital C and small c is crossed with capital C and small c, we get a capital C and small c. So cross it, capital C, capital C, capital C, capital C, small c, capital C, and both are small c. So what we are going to get? Both one must contain a capital C and another must contain a capital C. So what are here also total number of possible is 1 by 4, and this is how many times a capital C and small c is going to repeating? It is 2. 2 divided by 4 is 1 by 2. So we have predicted the number of chances of getting this capital A and what are the whatever product we want. But this is not going to the end of the problem. So how to apply this? We have multiplication or addition rule for further is important. So here we must apply, obtain the result by multiplication rule. Why multiplication rule means? as I told, or when we take the term individually, I took